So I'm joined by Jeremy Tinkler, and today we're going to be talking about ISO 10993. So to get started, Jeremy, what does the ISO standard 10993 do or aim to do? What's it aim to achieve?
Well, if you look at the scope of the standard, it's all clearly about biological safety. It's about safety assessment of medical device materials as they are used in the final form. And the scope talks basically about general principles. It says that you need to do your risk assessment because it's a risk assessment process. And that's inherent in the title. It's part of a risk assessment process. And it says you need to do this evaluation in line with the general principles, which are stated in clause 4 of ISO 10993 part 1. The rest of the scope of part 1 is about how to do the evaluation, about categorization of devices in terms of their of the way they contact humans, using existing data where possible, and doing a gap analysis to determine the procedure that you need to follow. So that's the basic scope as it's written in the introduction to part 1. And it does focus on these essential principles that are listed in clause 4. And this is because you can't really have a one-size-fits-all approach to biological safety. It's a complicated issue. So it's not just a list of testing. There are, in fact, no testing requirements at all in the ISO 10993 series. It tells you need to determine the nature of the assessment that's needed in line with the information you have and the situation that you find. So because it's entirely dependent on the nature of the materials and the nature of the contact. So these principles tell you to do it as part of a risk assessment process. There are basically two types of analysis. You're looking at a toxicological assessment, toxicological risk assessment, where you identify toxicological hazards and ensure that, because those come from chemicals, the toxicity is a result of exposure to toxic chemicals. So you look for toxicological hazards and you estimate the risk in line with ISO 14971 and in line with the process that's described in 10993 and the various parts of that. So parts 18 and 17 of ISO 10993 take you through this idea of identifying toxicological hazards and evaluating those risks. And the other avenue that you need to consider is biocompatibility as opposed to toxicological risk. Biocompatibility is the achievement of an appropriate host response on contact with a material. So this is particularly important, say, for implants and for blood compatibility. So you're looking at tissue and blood responses. So that takes you down a different avenue to the direct toxicological risk assessment approach. So you do it in two different ways, in essence, depending on the materials and the nature of contact. So to do the evaluation, what is required is a gap analysis. So you look at the existing data, and you need to match the existing data to the risk assessment needs. So mostly the discussion is on relevant data sources in the standard. I mean, there are 20 odd parts of the standard, and most of those talk about different types of data that you would need to fill those gaps that you have identified in your biological evaluation plan. So you need to see those testing requirements that are set out in the different parts of the standard. Those methodological requirements, you have to see them in the context of an overall evaluation that looks at a gap analysis based on the risk assessment process.
So, Jeremy, what are the main challenges or common mistakes that device manufacturers make when it comes to following the standard and conducting biological evaluations?
Actually, if you look back at the standard, it's always been misinterpreted. I think that's the most difficult issue that as standards writers we've had to contend with over the years. There have been continuous efforts for many years to prevent misinterpretation. So if you go back to before the 10993 series started, the initial standards in this area were based on work that was designed to look at biological safety of novel materials. And so they set out a test program by which you could investigate toxicity or biocompatibility of novel materials. And those that way of doing things was introduced into 10993 because it basically was built on pre-existing standards from the UK and the USA. And so the idea came about that in order to do a biological evaluation, you had to follow this recipe of testing. So for many years, there was an effort to revise Part 1 in particular to overcome this misconception, such that the 2018 version of the standard, the current version, emphasizes the need for chemical and physical information in all cases, because that's where the risk assessment starts. You start from the materials, you start from the classification of the device in terms of patient contact, and from then you do the gap analysis I've just been talking about to determine what testing, what additional information, data sets we call them, what additional information is needed for an evaluation. So this misconception has largely been dealt with by the 2018 version, although people still like to think that it's nice and easy. If you see a matrix and you say, oh, those are the tests I must do, you do the tests and you've ticked the boxes. But it's certainly not a box ticking approach. It requires an understanding, and that's another misconception. It's that anybody can do this. I mean, there are aspects of the evaluation that are fairly straightforward, but others require specific expertise. And there's a requirement in Clause 4 of Part 1, which is the general requirements, the general principles that I've mentioned, that the evaluation should be planned, carried out, and documented by knowledgeable and experienced professionals. So what does that mean? I don't think there's a clear idea about what that actually means, but it is an important requirement, particularly for some aspects of an evaluation. And regulatory bodies, and I've had, of course, some experience of this, have had many examples of these sort of problems arising where the problems in the field have resulted from the original assessment being done by people that don't actually understand what they're doing. And we've seen devices taken off the market because the hazards that were inherent in the device were not appreciated. So we've seen that with things like silver used as an antimicrobial agent in an inappropriate situation. We've seen aluminium leaching out of a device and causing high levels of aluminium exposure. So it's clear that oversight from people who understand the toxicity and the other aspects that are important, material science and so on, that's all important. So that's another area that is important. But the necessary knowledge is a scarce resource. So that's a new issue. Well, it's not really new, but it's another issue that is a big challenge. That is that there's not enough people who know the subject in order to provide the resource that is needed to regulatory bodies and manufacturers to make this, to do the right thing. So one other misconception, I mentioned that the fact that people think of it as a recipe for testing. With the 2018 version of the standard, some people took the idea that physical and biological information was required in all cases as an indication that you had to do analytical chemistry for every single device, which is certainly not the case. So that's another misconception that grew up in 2018 or following 2018. And this has been addressed by the 2020 version of Part 18, chemical characterization, and it's specifically within the risk assessment process. And that emphasizes the need for a stepwise approach where you first of all look at your existing information and see what else you need to do this gap analysis. And so that misconception has been addressed. So we just seem to move from one misconception to another. And I'm waiting to see what the next one will be.
So, Jeremy, you've mentioned a few times then that the standards changed over time. I mean, how has it changed recently? What are the main changes that people should be aware of and should be considering when they're following the standard?
Okay. Well, I mentioned the 2018 version of Part 1, which was quite a major event, really. It's state of the art. So since then, I think there have been two major changes that address the risk management process, and that's in Part 18 and Part 17. So Part 18 is on chemical characterization within a risk assessment process. And as I mentioned, it introduces this stepwise approach where you gather the information and then you generate new information when necessary. And that's a principle that should apply throughout the process. So this also clarifies when chemical analysis of extracts is needed, because it's not needed in all occasions. It's got more precise information on test methods and explains biological equivalence, which is an important area, particularly for clinical evaluation, where the European medical device regulation requires equivalence to be shown in certain circumstances, and is very particular about how you can demonstrate equivalence between different devices, but is lacking in any detail on biological equivalence. The understanding of technical and clinical equivalence is quite clear, but there's a lot of doubt over what biological equivalence means. And that has been explained in an annex to Part 18, where there's basically a definition of how you can demonstrate biological equivalence. So that's quite a useful step forward. Ongoing now is the is Part 17 revision. That's approaching the final stages of the drafting process. Maybe another year or so before that comes out. And that has, again, it's a name change, emphasizing the risk assessment approach, because it was a standard on allowable limits for leachable substances, and that was last revised in 2002. So since then, there's been a lot of advances in toxicological practice, and now the standard is being called the Standard on Toxicological Risk Assessment, which is a more accurate description of what it does. So one of the things it does, apart from providing more information on the toxicological processes and more recent ways of working in toxicology, it aligns with ISO 14971, which is important for any of these standards. They're all based on the risk management standard. And so it's important to understand how what we're doing in biological evaluation fits into the overall risk assessment methods and processes. And for toxicology, it's not straightforward, because there's a slight difference in the way things are dealt with between the principles in 14971 and the practice in toxicology. They're all based on the same principles. And it's similar with clinical evaluation, that the same principles apply as in 14971, but it's got to be interpreted slightly different. That the way you do it is slightly different, but the principles are the same. So implications from these changes, which are all based on risk assessment, this clearly has implications for part one. And it's being recognized that we need to take another look at part one now. Although there was a lot of clarification in 2018 and the standard has been very well received since then, it provided the emphasis on data sets and risk management rather than testing. But it's felt that we can go further in this area, and we've had a recent proposal from Australia, which has caused quite a stir, that suggests that we need to continue the process of optimizing the standard that is part one. Right.
So, Jeremy, does following standard to the letter result in a compliant product?
Well, in theory, any standard results in a compliant product if you follow it to the letter. But I don't know what letter it is you could follow for 10993. It's not really written in that sort of way. You need to follow the principles to the letter. You need to comply with ISO 14971. You need to comply with the principles that are in clause 4 of 10993 part 1. You need to do a gap analysis based on the existing data and then come up with a biological evaluation plan. So those are very clear requirements in the standard. So those you can say are complying with the letter. And people are getting used now to having a biological evaluation plan, but that has not always been the case. People basically didn't do it. Years ago, people didn't bother too much with clinical evaluation plans. Now you can't get away with that one. ISO 14971 says that risk management activities have to be planned. And so it applies to all of these standards. So you need a biological evaluation plan, just as you need a clinical evaluation plan and a risk evaluation plan. In fact, they're all part of the risk evaluation plan. Then you need thoughtful and scientifically robust selection of the necessary data sets that you're going to need following your gap analysis. And that identifies what sort of tests you may need to do. And of course, you've got to involve your knowledgeable and experienced professionals that I mentioned earlier on in the whole process. So those really, we're coming back to the basic principles, the risk management principles, the conduct principles. Just following those principles is basically how you demonstrate compliance.
So, Jeremy, just to summarize then, what would be your top 3 sort of bits of advice, best advice for people who are following the standard?
Well, these are the principles that we've discussed. They're, gather the information together and do your gap analysis. So start from where you are as early as possible, and that's your biological evaluation comes. Biological evaluation plan comes from the gap analysis. So that's the first thing. Start early, do that biological evaluation plan based on the existing data. Then be careful about how you define your data gaps and what data sets you need to evaluate those, because there are pitfalls in some of the areas that people are working on. Sometimes you get unexpected results. You quite often get unexpected results or unhelpful results in chemical characterization and implantation studies. So it's always good to think carefully about what you need to do. And there's nothing worse than finding a problematic results from a study, and then realizing that you shouldn't have done that study in the first place because it wasn't necessary. So, and then when you're analyzing your data, keep an eye on what it is you're looking for, because you need to define your acceptability criteria. That's a part of risk management, that you need to define acceptability criteria in advance. So when you've done your analysis and you've got all your data together, does that match the acceptability criteria that are relevant to the product and the way it's used.